Hello everyone, welcome to another new class of human physiology. In this week we were discussing about the skeletal system. So in last few classes we discussed about different type of human bones. We saw also about human joints. In this class we will discuss about human muscles. Also we will see what are different type of muscles and different components of muscle and how they work. So let's stay with it. So what different concepts will be covered for this class? We'll first see what is muscle. Then we'll discuss about types of muscle. Then we'll see structure of muscle fiber, filaments of muscle fiber, different crucial proteins which are present in the muscle, and also we'll little bit briefly discuss about smooth muscle and cardiac muscle. So let's go one by one. First, what is muscle? Muscle is defined as a tissue primarily composed of specialized cells, fibers, which can contract to effect movement. So basically, as we all know, if there is certain environmental responses, for example sensory responses, those responses generally are accepted by the sensory nerves, and those signals are sent to first spinal cords and then from spinal cords to brain. Now in response to those sensory responses, many a times brain sends the motor signals via the motor nerves, and these signals reaches to different type of muscles, including skeletal muscle, smooth muscle or organ muscles, and then these muscle cells performs action, and in terms of those initial sensory responses our body kind of performs certain reaction. So in human body basically we have more than 600 muscles, and as we already said, the main primary function of muscle is the transformation of the chemical energy into the mechanical energy. And why the mechanical energy is needed? Basically it is needed to generate force, perform work and produce movement. Apart from that, muscle tissues also stabilizes our body's position. It also regulates different type of organ volume. It also generates heat and maintain overall heat balance in our body. Lastly, it also propel fluids and food matter through different body system. And the scientific study of the muscle is also known as myology. So what are different type of muscles? So as you can see in the images, we have mainly two type of muscle. One is striated muscles. So in the striated muscles you will see this type of striation, right? This type of striation. So these are called as striated muscle, and it can be either skeletal muscle or cardiac muscle. And secondly, there can be also non-striated muscle. For example, you can see in the smooth muscle, it is non-striated muscle because there is no striation present. Depending upon the control, the muscles are also classified into two types. One is voluntary and second is involuntary. For example, in case of voluntary, this muscle can be controlled by the will, like skeletal muscle. In cases of involuntary muscle, which are present mostly in terms of organs, these muscles cannot be controlled as per will, and it can actually perform without any specific stimulation also. Mostly these types of muscles are innervated by autonomic nervuses. Let's see with the first one, which is the skeletal muscle. So this is a gross overall structure of the skeletal muscle, which is basically situated in association with the bone. So you can see this is a bone over here, and from the bone first there is the tendon. So muscle fibers are also connected to the bone via the tendon, and as we know tendon is a connective strong tissues. Now as you can see, right from the tendon we have layers and layers of muscle fiber which are basically formed together, and each of these muscle fiber has a specific membrane on top of it. So from the bones and with the tendon, the first coil of this muscle fiber what is coming out, the top membrane of it is called as epimysium. And from the epimysium, if you can see that there are this lot of round kind of shape which are basically called as fascicles. These are like bunch of like muscle fibers together, which is called as fascicles. So if you basically pull up, so if you basically like simply pull out one fascicles outside, so epimysium is the outer layer, and if you pull out the one like bundle of like myofibrils or a bundle of like fascicles, the membrane of it is called as perimysium. Okay, so it is basically 3 layer of membrane which are present in the muscle. The outermost layer, which is the epimysium, this is the outermost layer of the muscle fiber. And now if you pull out one group of muscle, then this middle layer, which is called as perimysium, and then you can see further there are small fascicles. So one fascicle if you pull out, so if you pull out on single fascicles, you will find like the unit cell of the muscle or like myofibril. Inside of the myofibril, this layer or the membrane layer, which is called as endomysium. So basically we will have like 3 layer. The outermost layer is called as epimysium. Then we have the middle layer, which is called as perimysium, and just the above of this myofibril, which is also kind of unit of muscle cells, you can have the most inner layer of the muscle, which is called as endomysium. Okay, so this is basically the 3 layer of muscle membrane. And now inside of that myofibril or the muscle fiber, if you see that each muscle cell is basically cylindrical in shape. So you can see that each muscle cell is kind of cylindrical in type of shape, and the average length of the fiber is about 3 centimeter. Next, further you can see that each muscle fiber is enclosed by a cell membrane called sarcolemma, and this sarcolemma is just above the endomysium. So you remember in the last slide also already we discussed that what is endomysium, which is basically, so if it was like the myofibril, so the upper part of the inner layer of the myofibril was the inner layer of the myofibril was called as the endomysium, right? Endomysium. And now just beneath the endomysium there is another cell membrane which is called as sarcolemma. Cytoplasm of the muscle is also called as sarcoplasm, and inside of the sarcoplasm there are a lot of endoplasmic reticulum, which is called as sarcoplasmic reticulum. And the role of sarcoplasmic reticulum is very crucial because it stores the calcium, and during the contraction process from the sarcoplasmic reticulum it releases the calcium and the muscle contraction happens. So how sarcoplasmic reticulum it releases the calcium, that we will discuss in our next class, but just try to remember these are like some basic components of the muscle fiber. And also importantly, myofibrils are the fine parallel filaments present in the sarcoplasm of the muscle cell. So myofibrils are very important. This is the fine parallel filaments present in the sarcoplasmic muscle cell. Now let us see what are those filaments of the muscle fiber. So basically muscle fiber consists of 2 type of filament. One is the thin filament or actin filament, and the second is the thick filament or the myosin filament. So in cases of thin filament or actin filament, these are mostly anchored to the Z disc. What is Z disc we will discuss very soon, but just consider that these thin filaments are anchored or attached to the Z disc. And in the thin filament you can see different type of protein. The most primary or important protein you can see is the actin. So actin is basically one of the main or key component of the thin filament, and individual actin molecules you can see it basically kind of creates like a helical, like a twisted helical kind of filament formation. So this individual actin molecule eventually it creates like a helical twist. There are two more protein, one is troponin and another is the tropomyosin. So we will discuss about all of their functions in the next slide. In cases of the thick filament, myosin is the main component of the protein, and in skeletal muscle almost like around 300 molecules of myosin form a single thick filament. So 300 myosin molecules of myosin basically form this single thick filament. And as you can see, once like this myosin thick filament kind of gets twisted for a thick filament, it also gives like a golf bat like structure. So after the twist, it kind of gives like a golf bat like structure, and this part where this looks like a golf bat head or golf bat like structure, it is called as myosin head. Let us see their function. So in cases of myosin, which is present in case of like thick filament, right? So this is basically a contractile protein, and the myosin molecules contains like both tail and two myosin head. So it basically has what, like as we said, it basically kind of creates a folding. So it has a head, like it has two head, and then it also has a tail. So basically myosin consists both tail and head. This binds to the myosin binding site on the actin molecules of the thin filament. So basically what it does, that these components of myosin, it binds to the myosin binding site of the actin molecules that is present in the thin filament. And when it happens during the muscle contraction. We will see in next slide how it exactly happens. Then as we said that in cases of thin filament there are three proteins. So myosin, here if you see, in cases of myosin this is present in the thick filament, right? And then actin, tropomyosin and troponin, these are present in case of thin filament. Okay. So actin is also like a contractile protein. This is the primary protein present in the thin filament. On each actin molecule there is a myosin binding site. Okay. So basically each actin, so if you consider like actin kind of protein, it has like a myosin binding site. Okay. So it has like a myosin binding site. Okay. So basically each actin protein has a myosin binding site where myosin head of the thick filament can bind, right? So this receptor or this protein binding site is called myosin binding site. Okay. And in this myosin binding site, what happens, this head part of the myosin can eventually bind with the actin. Okay. The next is the tropomyosin, which is a regulatory protein present in the thin filament. When a skeletal muscle fiber is relaxed, basically tropomyosin covers the myosin binding site. So what we are saying, that in case of the thin filament, for example we have this actin molecule, and during relaxation, let us assume this tropomyosin is bounded with the myosin binding site which is present on the actin. And if this tropomyosin is already bounded in the actin like with the myosin binding site, what will happen? Myosin will not be able to come and bind, right? So basically myosin will not be able to come and bind to this myosin binding site of the actin. So this troponin, this is tropomyosin, which is basically a regulatory type of protein that eventually blocks the binding site of the myosin to the actin. And then the last one is the troponin, which is also a regulatory type of protein present in the thin filament. And what happens basically during the muscle contraction, whenever this calcium ion Basically, whenever there is a release of calcium ion from the sarcoplasmic reticulum, this calcium ion binds to the troponin. So, what happens is like basically with the troponin, calcium ion binds and then it undergoes like a shape and conformational changes, and eventually it removes this tropomyosin away. Okay. So, what basically the all 4 protein we discussed first about myosin, which is the part of thick filament, and in case of myosin we saw that it has like both head and tail. Head mostly binds with the actin in the myosin binding site. Actin is present in the thin filament, and now it has 2 regulatory protein which are present in thin filament. One is tropomyosin, second is troponin. When muscle is at relaxed condition, basically with this myosin binding site, tropomyosin gets attached, and that prevents any this head groups of myosin to come closer and attach with it. But when during like the muscle contraction, when the sarcoplasmic reticulum releases the calcium, this calcium binds with the troponin, causing like a conformational changes of the protein, and eventually what that does is it removes this tropomyosin away. So, basically it removes the tropomyosin away from the myosin binding site, leaving the open ligand binding kind of site inside outside of the actin, and in that case myosin can easily come and bind to the actin, causing like muscle contraction. So, hopefully you kind of understood different type of proteins which are present in the muscle. Now, let us quickly see like how this muscle contraction happens and what are different roles of the protein. What we just said we will just go through in a pictorial way. So, basically you can see this is the thick filament, right. So, in the thick filament it has basically like 2 type of heads, 2 heads we discussed, right. It has like this type of structure where there is this twisting, right, twisting of like lot of myosin protein, but eventually it creates like a 2 golf bat like heads, right, 2 golf bat like heads. So, you can see that in the first step, whenever there is a release of calcium, and how it generates we will discuss in the next class, but just remember that neuromuscular transmission happens. During the process of neuromuscular transmission, calcium is basically released by the sarcoplasmic reticulum. So, during the neuromuscular transmission, calcium is basically released by the sarcoplasmic reticulum, and you see that this calcium, what it does, already we said this calcium binds to the troponin and causing like a conformational change as shape of this myosin head, right. So, basically troponin, when it binds, it removes those tropomyosin away. It removes those tropomyosin away which are the already occupying those myosin binding site. So, that means myosin can bind now to the actin, right. So, once calcium comes in, one more time let us recall, once calcium comes in, it binds to the troponin, troponin removes the tropomyosin away from the myosin binding site. That means myosin can bind to the actin, and now once myosin can bind to the actin, you can see the bound myosin rotates its head. So, basically a rotation of the head happens, producing a power stroke. In the next stage, like ADP is released and ATP molecule binds with the myosin. So, basically from this condition ADP is getting released and one ATP molecule is bind to the myosin head. In the next step what happens, you can see both this actin part and the myosin protein part, they get detached and ATP is hydrolyzed. So, basically 2 things happens, that actin and myosin both gets detached from each other. Next step, ATP is hydrolyzed. Again ADP bound myosin head is cocked and ready to bind to the actin again. So, basically in the next step again these rotational changes happen to the head, leaving the myosin to be bound again to the actin for the next cycle whenever there is calcium ion comes in during the process of neuromuscular transmission. Okay, so this is the way like both actin and myosin kind of work together. Also with two regulatory protein that is present in the thin filament, troponin and tropomyosin. There are some other important proteins also related to muscle. For example, titin, which is like a structural protein that connects a Z disc to the M line of the sarcomere. Then actinin, which is also a structural protein of the Z disc that attaches to the actin molecule. Then nebulin, which is also a structural protein that wraps around the entire length of the each thin filament. Finally, the dystrophin, which is a structural protein that links the thin filaments of the sarcomere to integral membrane proteins in the sarcolemma. This is attached in turn to proteins in the connective tissue matrix that surrounds the muscle fiber. So these are some additional protein components that has important role for overall muscle. Now let's see, as we are discussing like different like Z disc, M line. So what are these, right? So basically if we check the microscopic structure of muscle or the muscle fiber, where sarcomere is generally called as the unit cell or the functional unit of the skeletal muscle. So what is the functional unit of the skeletal muscle is the sarcomere. So this is basically a structure of a sarcomere, right? And each sarcomere is basically extends between like two Z lines of myofibril. So in the myofibril, so you remember like we discussed like about myofibril, right? We discussed about myofibril. So in the myofibril you will see like this type of unit cell which are called as sarcomere. So this one cell which is called as sarcomere, these are functional unit of the skeletal muscle cell. And each sarcomere basically extends between two Z lines. So if we think like that, each sarcomere is basically extend between two Z lines. So you can see here these are the Z line or the Z disc. So this is, and this part is called a Z line and the Z disc, and exactly in between we have like one sarcomere unit. Okay, and in the sarcomere you can see like both thick and thin filament. So this part you can see all the thick filament components are there, and you can see like thin filaments which are the actin, troponin and the tropomyosin. This has been mostly here, but there are certain thin filament which is also infused with the myosin or with the thick filament. So basically this part we have the thin filament, right? Some amount of thin filament is also present with the thick filament. So basically you can see from this part, the start like wherever the thick filament has been started, from one corner to the another corner of the thick filament, this is called as A band. So what is basically A band? A band is completely the total zone or the region of the thick filament, although in the corners there might be some thin filament also that is also infused with the thick filament. So this is called the A band. Okay, so basically A band is the complete zone of the thick filament. Now in the corner you see like this is only thin filament, right? There is no presence of any thick filament. So this component or this part is called as I band. So you will have two I band in the both side, left side and right side, where there is no presence of any thick filament. That means only actin, troponin, right? So only actin, troponin and tropomyosin, which are part of the thin filament, is present. So this part is called as I band, and then in this area especially there is no presence of any thick filament. Okay, so A band is the total zone or area of the thick filament, which is mostly the myosin, but in the corner area there is also infusion of thin filament also, which are like actin, troponin and tropomyosin. In the corner area just beside the Z disc, you can see only the thin filament is present. This is called as I band. Now in between, you see in between this part basically, where there is no presence of any thin filament but only thick filament is there, it is called as H zone, and this H zone is like very dark dense area, like a darker area. You can see there is no presence of any thin filament here, so only pure myosin components will be present in this middle area, which is called as H zone. Inside H zone there is also a M line. Basically M line is the center of the H zone that contains certain protein to hold the thick filaments together at the center of the sarcomere. So M line is basically the center of the H zone. Okay, so you can also kind of go through all these like Z disc, which is basically the narrow plate shaped regions of the dense material that separate one sarcomere from the next. Then A band already we discussed, which is the total region of the thick filament. But in the in the A band, in the mostly the adjacent to the I band, you will also see there is a overlap that happens in the thin filament. But I band on the corner near to the Z disc, those are purely the thin filaments. There is no overlapping region. And in the middle part there is only thick filament, there is no overlapping with the thin filament. That particular dark grey area where only thick filaments are present is called H zone, and the middle line of the H zone is called as M line. Okay, so go through those, hopefully you are basically you understood that basic component and structure of muscle. Then lastly we will discuss about smooth muscle, which are non-striated cells. So you can see like it can be with binuclei non-striated type of cells, and these can be seen in different area. For example, like wall of organs like esophagus, stomach, intestine, ducts of the digestive gland, trachea, bronchial tube, urethra, urinary bladder, wall of different blood vessels, mammary glands, and ciliary body of the eye. You can see so many area, you can see this smooth muscle cells are present. Then these fibers are basically generally very small, almost the diameter is like 2 to 5 micron, and in the length it is about like 50 to 200 micron. Nucleus is single and elongated, mostly it is present centrally, but generally 2 to more nuclei can be seen inside of the nucleus. So you can see 2 to 3 nuclei can be present inside of the nucleus. And in terms of type, smooth muscle can be either single unit smooth muscles, which in case of single unit smooth muscles, you can see the fibers that connect to one and like the other, it is by gap junction. So basically multiple fibers can get connected to create like a single unit smooth muscles, and they are connected by gap junctions. And this way they form like a network of smooth muscle. Where it can be seen, it can be found in almost every small artery and veins. Also in case of like different hollow organs such as like stomach, intestine, you can see the single unit smooth muscle. And in case of multi-unit smooth muscle, you will not see any this type of gap junctions or network of smooth muscle. Mostly this can be found in the large walls of the large artery. Apart from that, it can also be seen in the lung, pupil diameter, in the ciliary body, etc. Finally, one of the most crucial muscle, which is like the heart. So heart is also called, as you know, as a muscle, but heart is, as you know, it is a striated type of muscle. The principal tissue in the heart wall is a cardiac muscle, and the cardiac muscle fibers have the same arrangement of actin and myosin, what we discussed already. However, there are presence of this intercalated disc. So you can see like the intercalated disc, this is different from like normal skeletal muscle. So heart has a presence of this intercalated disc. These are unique to the cardiac muscle fiber. These microscopic structures are generally irregular transverse thickening of the sarcolemma that basically connects the end of the cardiac muscle fiber to one another. This disc contains desmosomes that hold the fibers together and the gap junctions. This also allows muscle action potentials to spread from one cardiac muscle fiber to another. Also, it is very important to kind of highlight that in the response of a single action potential, generally cardiac muscles remains contracted almost 10 to 15 times longer than the skeletal muscle. Okay, it is due to like the prolonged delivery of the calcium ions to the sarcoplasm. So basically more amount of and the prolonged delivery of the calcium ion cause like more than 10 to 15 folds of prolonged contraction in a single action potential in case of a cardiac muscle. That is different also from the normal skeletal muscle. So if you like the class of muscles, so do you know like cardiac muscle contracts continuously without any stop. Cardiac muscle tissues contracts and relaxes about 75 times a minute during resting condition. Cardiac muscle fibers can also use lactic acid produced by the skeletal muscle fibers to make ATP. So ATP is like all the time needed, as you saw like how ATPs were getting attached during the muscle contraction step and how the overall process happened. So it is also benefit during exercise. So during exercise, as you know, like lot of lactic acid is produced, and this skeletal muscle fiber can use this lactic acid to make more ATP. So activity can you also like answer this question that which type of smooth muscle is more like cardiac muscle than the skeletal muscle with respect to both its structure and function. Okay, so hopefully you are enjoying the classes of human physiology. In the next class we will discuss about like neuromuscular junction, and we will see like how the process of this neuromuscular junction happens and how the contraction, muscle contraction initiates. Okay, thank you again for attending one more class of human physiology. We will meet with you very soon with another new class. Thank you.